Hi everyone. This tutorial is intended to take you through writing your lab report. This is a skill I think is worth getting good at since it's continuously examined during this degree and any master's or PhD programs you might want to do in the future. Keep in mind that the tips and examples that I'm going to give you during this tutorial aren't perfect. I've just lifted them from my previous lab reports, but I have taken into consideration the feedback that I received so that you can avoid some of the mistakes that I made during my first and second year submissions. Before you even begin writing, one of the most important things to consider is what your research story is. This is to help make your report a narrative of your project. I'd like to think of this as if you were writing a story in English. So you'd start with the background of your project, which includes a disease related to it and previous research done in the field. You then move on to your hypothesis and how you've ended up choosing it. Following this, you can think about why you've done each experiment and how the experiments follow on from one another. Then think about what you can conclude from each of your experiments and what you can conclude from your project as a whole. I'd then also like to consider what your research contributes to the field. This really helps to make your report cohesive, so you're going to hear me say the phrase research story quite a lot during this tutorial. A good thing to remember is that even if you've collected the data as a group, your report is your own work where you must interpret the results individually. So feel free to create your own research story for your paper even if it differs from the other members of your team. Now, the first thing visible on your report is the title, meaning that it is quite important in framing your report. From previous feedback I've received is that the title must reflect the findings of your paper. An example I've just made up is knocking out WIP1 increases the susceptibility of breast cancer cells to cisplatin. So, before you even read my imaginary paper, you have an idea of what it's going to show. However, I've been through LabPod research, it's entirely possible that your results aren't this conclusive. In this case, it isn't a problem. I would suggest using words such as modulates, which are non-directional, but it still shows that there's some effect to what you're investigating. Conversely, negative results are still valid, so you can just say that the thing you were investigating does not have any effect. You then have to list the authors of the paper. List yourself first, and the other members of your LabPod group in order of their surnames. You must also remember to add affiliations as this carries marks. I've given an example of one way to do this here. At this stage, you're all likely to have the same affiliation. Now, the first section of your body of the text should be the abstract. This is a fast-forward look through your whole paper. As you know, abstracts tend to be quite short with very strict word limits, meaning that every sentence counts, so you must be careful to be brief but informative, keeping in mind your research story. A rough template I've used in the past when writing abstracts is this. First, I add a sentence about the background of the disease with some statistics to show why this research work is important. Next, I introduce my target proteins and pathways and explain how they link to the disease. From that, I explain what the study is investigating and why, basically my hypothesis. Following this, I add a sentence about the main finding and conclude with the translational potential of the study, meaning what is the contribution of your study to the bigger picture of the disease. Another thing to remember is that abstracts are meant to be standalone, so remember to put terms in their full form before abbreviating them, and make sure that if someone else is reading it, they can understand it without reading the rest of your text. Another thing to note is that abstracts don't typically have references, so don't put them in there. It probably goes without saying, but try to write this section last after you've written the rest of your report and you have an idea of what it looks like. Now let's move on to the introduction. This section is very important to give the reader an idea of the background of your study and why you've chosen to investigate what you're investigating. Once again, this means it reflects your research story, so make sure you have an idea of what that is before beginning your report. To do a quick breakdown of what's required in this section, you should once again start with the disease background. They are looking for evidence of outside reading to back up your project decision, so make sure to add in a lot of references within this section. You can then move on to explain in more detail your target protein, pathway, or gene, and reference other studies that have contributed knowledge in this field, and explain how this all ties in with your project. Basically, explain the gap that your project is trying to fill. You can then state your hypothesis clearly in a couple of sentences, and finally, based on previous feedback, they really would like to see a concluding sentence describing what your findings show before moving on to the next section. A top tip I have to make it look like you've thought about your project more than you actually have is to create your own diagram for a graphical hypothesis. This is one I made for my LabPod 2 report that got quite good feedback from the markers. I made this one on PowerPoint, but there are more tailored programs out there. One off the top of my head is BioRender, which really allows you to make nice biological diagrams, and it's quite a user-friendly and free interface. You can also add in elements showing your findings and make this a graphical abstract. I'd suggest looking for other research papers that do this in order to get ideas for how you can do yours. Next, let's move on to the methods section, which is notorious for eating through your word count if you're not careful. You must be able to balance brevity and details within the section, which is quite a daunting task if you've never done it before. The easiest way to get a hang of this is to read other papers, since the methods that you've used, cell culture, Western blotting, PCR, they've all been done before. So read a few papers and you can compare 2 or 3 of them to look at what the important details are, because these would be the recurring details in every paper. Off the top of my head, some of the most important details are the product names if you've been using a kit, a brief recipe of the buffers if you've made them yourself, and time points if you've done treatments for your experiments. For Western blotting immunostaining, some important details are the antibody supplier, catalogue number, dilution, and whether they were raised in mouse or rabbit or any other animal. And for PCR or transfections, mention the primer and gRNA sequences. A top tip for saving words is to include this information in tables within an appendix. Just mention in the method section that it can be found in Appendix 1, for example. I've included an example here of a method section that I've done in one of my reports. It's not perfect, but it does include how to add in buffer recipes and concentrations of reagents used. You can pause the video here if you'd like a better look at it. After this, we have the results section, where you can display all the data that you've collected across your project. It's important to remember that this isn't your discussion, so just present your results in the text. Don't explain them beyond what can be seen in the figure or discuss how they link into each other just yet. I would recommend using subheadings in this section, such as determining the optimal drug concentration, just to break up your results section a bit and allow the reader to follow through with you. Start by describing why you're performing each experiment, so basically what the aim of the experiment is. And also remember to refer to the figure numbers within the body of the text. So something like the Western blot results in figure 3A show that, whatever. This just helps keep your reader informed, especially when formatting doesn't allow your figure to be right next to the corresponding text. Also, again, with regards to figures, make sure they're clearly labeled and keep your legends informative but concise. Figures are meant to be standalone in papers, so in the legend, make sure you describe what the experiment was, the conditions you've used, and any statistical analysis you've used as well. Don't be tempted to interpret the results within the legend, as that's not appropriate. I'm including examples of the results section and corresponding figure from one of my reports that I got decent feedback for. Again, you can pause the video to get a better idea of this if you need. Now, let's move on to what is arguably the most important section, so your discussion and conclusions, because this allows you to showcase your own scientific interpretation of your results and really draws in on your understanding of the project. A good way to start this section, keeping in mind the flow of your report, is to remind your reader what the purpose of the study was in the first couple of sentences. After that, it's quite nice to explain the results chronologically in terms of whether what you found was expected, or you can try and explain why the result was unexpected. You can then also link in your results. Do they back each other up or do they disagree with each other? You can then reference other papers that either agree with or disagree with your findings and point out how this affects your understanding of your results. Are they reliable? Are they unreliable? What would you do in the future? The teaching fellows also like when you reflect on your own project, like how has your project contributed to the knowledge gaps, or how could it eventually contribute to them, and what assays do you think you can perform if you could do this experiment for a longer period of time. Finally, you can state the overall conclusion of your results and link this back to the disease and what this could mean for patients, etc. So basically your translational potential. Again, there's no defined recipe for a discussion, so there might be things you want to add or remove from this, but I think this is quite a good framework to get you thinking about it before you begin. To close things off, let's look at the take-home messages of this tutorial. I want to stress how important it is to read papers, as it gives you an idea of the tone that they've used, the details that they've included, and the way that they interpret results. Secondly, let's talk about how to deal with negative results. Lab pods aren't about stellar results, they're meant to teach you how to interpret the results that you have and to show your understanding of lab research. So don't be afraid to include your negative results and state why it went wrong, and you can show your ability to troubleshoot the experiment. Next, the phrase you're probably tired of hearing by this point, your research story, which is integral to the flow of your report and allows you to develop an idea of your paper before you even begin writing it. Finally, remember that this is a scientific piece of writing, so although I've been banging on about how you need to make it narrative, don't write it as if you'd write a novel. Avoid using words like I or we, and try to keep your tone as formal as possible. Reading papers in high-impact journals such as Nature or Cell will give you an idea of what is required. I hope this tutorial has been useful for you. We hope to hold a question and answer session if you require, so please message us with feedback. Good luck with your report writing. Bye.